get just world famous paper cited hundreds and hundreds of times. Especially remarkable because it's
Q&A, make sure you get him.
sort of specific area that's showing how critical it is to understanding processes at all levels, from cells to whole plants to whole ecosystems. It is apparently got into studying plants at the age of 12. I found out last night. Give him a step up for sure. He did his undergraduate at Duke. He did his PhD at Harvard with Martin Zimmerman, just absolutely exceptional hero. People say the god of hydraulics, and then he did his postdoc with Mel Tyree, who would be then considered the Abraham of hydraulics. And then it's been at Utah, then briefly at Duke, and then back at Utah. John Sperry is especially famous for refining and developing a deep understanding of the air seeding hypothesis, which is basically the way that wood loses function as trees are droughted. And then the method to quantify that, the vulnerability curve, get details of drought tolerance, how plants respond to freezing, the meaning, the deep significance of wood density, and then looking at scaling relations at all levels, combining very rigorous experimentation with cutting-edge theory, statistical theory, and modeling. So for all these reasons, very inspiring person.
Well, thank you, Warren. Very nice introduction. And thank you all for coming and listening to me enthuse about my favorite tissue, which is the xylem. So I'm going to tell you about why I'm so interested in it and try and make it as interesting as possible for all of you. The first thing I want to mention about the xylem is, of course, it's transporting water, and this is a cross section of it, and all the little tubes in here that are lit up are the xylem pipes. And the first thing I want to tell you about that is how much water these pipes move, because it turns out to be significant. Here's my mom here. My mom. We don't have any kids, so I have to show pictures of my mom. But a tree of the width of my mother's waist would be transporting about 26 gallons of water a day. So trees and plants in general are very thirsty organisms. And if you sum this water use up through all the trunks of all the trees in a watershed, there's just about as much or more water moving through these pipes in this collective xylem stream as there is in the streams that drain that watershed. So the plants are in the business of moving lots of water through those tubes. Why are they doing this impressive feat of transport? Well, when I ask students, why do you have to water your plants? They always say, they always say, well, it's photosynthesis because the water is consumed in the photosynthetic reaction, and they're disappointed to learn that less than 1% of that water is actually donating electrons and being consumed in the reaction. Then they next go, well, cells are 98% water, so in order for the plant to grow, they need water to fill the cells. True enough, quantitatively less than 5%. Most of this water, over 95%, is evaporated through open small pores in the leaves. And plants, not surprisingly, don't have open pores in their leaves just to show off how much water they remove. They have them in order to take up CO2 efficiently through the atmosphere. And so you have this inevitable exchange of water for carbon. And the exchange rate is normally a very poor one for the plant because the diffusive gradient for water loss is so much greater than the CO2 uptake under most conditions that you have a river of water exchanged for relative trickle of sugar. And so this is why plants use so much water, and because they need so much of it, that's why it's limiting for plants. It's a limiting resource. If plants could figure out how to take up CO2 without losing so much water at the same time, water would be using about 5% of their water budget. Water would not be a limiting resource for plants, and places like this would look a lot different, presumably. So what makes this river of water flow? How does the plant transport all this water? It has to do it very efficiently, or you can imagine it would quickly starve to that. It had to pump this water with energy, since the exchange rate is so poor, it run out of energy and starve. And indeed, the mechanism that has evolved to move this water is incredibly efficient. It's a wick. The water is wicked up, and so it's a passive process. The physics, the only important, well, not the only, but for now, important physics about this wicking process can be summarized in this very simple diagram. The evaporation in a wick is from the air-water interface. And that interface, that meniscus, is anchored by capillary forces. These are the adhesion of the water molecules to the hydrophilic wall of the wick and the surface tension that wants to keep that meniscus flat. So as water is evaporating from that meniscus and it wants to curve because the water is moving, it doesn't curve because these forces oppose that, and they exert a subatmospheric pressure on the liquid water behind the meniscus, which pulls water from the source through the water-filled pore spaces of the wick to the evaporating surface. So you have basically bulk flow of water down a subatmospheric gradient in pressure that moves water to the site of a stationary meniscus that's anchored by capillary force. That's how it happens. And so there's no metabolic energy that is required to drive the mechanism. Now, of course, the wick is very complex. The structure of the wick has the evaporating surface in very small nanometer scale cell wall pores in the leaf. And that's where the actual capillary suction is generated. And the significance of the pore size is the narrower those pore spaces are, the greater the capillary suction. That suction is coupled to bulk flow in much wider tubes. These are the xylem conduits. They're on the order of 20 to 200 microns. Several orders of magnitude larger than the sucking pores. And of course, the significance of that is to minimize the flow resistance in the bulk flow phase. Down here at the bottom of the wick, water is filtered across a osmotic membrane at the endodermis by reverse osmosis, which of course provides selective uptake. Now, an important component of the wick, as you can see, these tubes are compartmentalized. And the reason for them being compartmentalized is because they're so wide that they themselves, when they become air-filled, which they do, are unable to generate much of a wicking force because they're too wide. And so when they embolize, they stay gas-filled, and of course that's bad for the plant. And so they're compartmentalized, separated from other conduits by pitted end walls that keep this air from invading the rest of the wick. So these same capillary suction forces up here are replicated down here to trap the air-water interface. So that's pretty much how it works. There's not a living cell in this diagram. It's all physics, and that's beneficial to the plant because, again, it doesn't have to exert beyond growing the wick. It doesn't have to exert much metabolic energy to keep it going. The cells, I should mention, these xylem conduits themselves are dead at functional maturity, so they're just cell wall skeletons, no protoplasm. So it really is at heart a physical process. Living cells are important, though, because they regulate the flow. They don't drive it under transpiration conditions. They regulate it. They're capable of sending signals, hydraulic or otherwise, to these stomatal pores to modulate the flow in the system. And we'll come back to that. So you can summarize this cohesion-tension mechanism as a tug of war on a rope of water between capillary forces in the leaf cell wall pores and in the soil pore space. Very efficient as it has to be. But as amazingly efficient as it still is an important process that influences plant performance. And there are two key characteristics of this wick that propagate to influencing the performance of the plant. And that is the flow resistance through it, and then the strength of the water columns inside these conduits. And so in my talk, I'll first sort of talk about the implications of flow resistance, and then later come back to this strength element. Now, it's very easy to manipulate the flow resistance of the xylem and see what happens. You can knock out experimentally the conduits and increase the flow resistance. And we've done this experiment many times, but one of the nicer examples is this experiment that Ron Hubbard did with ponderosa pine, seedlings maybe about this fall in the lab, and we experimentally reduced the hydraulic conductance. I'm going to reciprocate from the resistance to conductance all through the talk, so you'll just have to bear with me there. So as we have a lower hydraulic conductance, you're increasing the flow resistance. And then we measured in a gas exchange chamber the stomata conductance. This is the diffusive conductance, the water vapor through the stomata openings. And what you can see is as we reduce the hydraulic conductance, the plant responds by closing the stomata, almost exactly proportionally. And the consequence of that is it's harder for the plant to get CO2, and so the photosynthetic rate goes down almost also in proportion. So there's this link between hydraulic conductance and stomatal conductance, which ends up constraining the productivity of the plant. Why should there be this link? There's no physical reason why there should be a link. The diffusive conductance is not in any way constrained by the flow resistance. It has to do with the living cells and how they're regulated as well. So what happens in this experiment is that you first increase the resistance, and the next thing that happens, okay, got the same amount of flow through here. The stomata don't care what's happening down here at this point. Same amount of flow, fewer conduits, so the pressure drops up here at the top of the wick. And the leaf cells sense that drop in pressure, and they send a signal, hydraulic or otherwise, to the stomatal apparatus, which then responds by closing. And the closure is what, of course, reduces the flow, because now we have less transpiration, and then that feeds back to mitigating the pressure. So you've got less flow because you have fewer conduits, so you have the same pressure. So you've got this sort of homeostatic negative feedback loop that's regulating the leaf, the canopy pressure. The plant is obviously deciding to regulate its pressure rather than continue photosynthesis, because the downside of this is the closing is wow. And we'll get back to why the plant might make that decision a little bit later. So the coupling between hydraulic conductance and photosynthesis in this ponderosa pine experiment is mediated by this leaf pressure regulation. And there are a lot of implications of that. One of which I've gotten involved with recently is, and I talked about this for longer than I should have probably yesterday, metabolic scaling theory. And this was first proposed by Jeff West, Jim Brown, Brian Inquist. And they used this principle to model the allometry of hydraulic conductance of trees with their mass, and thus predict the allometry of the productivity of trees with mass to get a metabolic scaling prediction. And the original results, still don't want to respect. The original results were very interesting because they predicted 3/4 power scaling of growth rate of plants, dry mass per year, versus their body mass. And for the vascular plants, you can ignore the algae down here, they don't have pipes, but for these guys, the model predicted and the observations seem to come close to that, a 3/4 power interspecific scaling. And this is based on a hydraulic model of the vascular system and the linkage with photosynthesis. Now, in the last few years, I've gotten involved in this interesting concept in a collaboration with Brian Inquist, one of the original proponents of the theory, and Van Savage here at UCLA, and Peter Reich at Minnesota, and members of our lab groups. And the goal of this, as I described it, but I'm just going over the 5-minute version for those who weren't there yesterday, is to integrate this pipe work, this internal pipe work, with the external branching network, and refine and better test this concept that you can predict from vascular properties, large-scale metabolic processes. My role in this has been to modify that original very simple hydraulic model, which was an analytical model, to a more complicated, but not a whole lot more, model, which is necessarily numerical in its solution. And to combine, again, it's got 12 parameters, I won't go over them, but basically specify the external branching network and specify the rules of the internal pipework. And the goal of this model is to predict, the specific goal is this network model is supposed to predict this equation between how much water the tree is moving and its size, in this case, basal diameter. So the goal of the model is to provide species-specific predictions of this holometry. And so we're testing it in 2 really different species. To challenge the model, we're testing it in plants called diffuse forest plants, which have a lot of narrow pipes dispersed over many growth rings, and maple species, and then a so-called rainforest species that has the water flowing through relatively few big pipes confined to a single very narrow growth ring. And when you measure the sap flow rate, this scaling that we're trying to predict, sap flow versus tree size, you can see there are major differences between these species. The maple is actually using about twice the water that the oak is in the same size, despite the fact that it's skinnier conduits. So we can get the 12 parameters for the model for these 2 species, and then see how well it predicts this scaling. And as I said yesterday, I was stunned to find how well it did. How well, like a 12-parameter model, based mostly on anatomy, could predict daily sap flow as a function of size. Maple, again, the model predicts not only the slopes of these scaling relationships, but the relative values of the species. So we can begin to use it then to predict and explain different rates of water use between plants. Now, going back to metabolic scaling theory, I should say, if you then the model will also predict sap flow as a function of tree mass. Okay, that's exponent is shown here. It's about 0.6-ish. And again, it agrees with measured exponents of flow rate by tree mass. So the model does a pretty good job of predicting flow allometry. Now, the main tenet of metabolic scaling theory is that from the flow allometry, if flow rate is proportional to growth rate, then from the flow allometry, you should get the growth allometry, the metabolic allometry. And to test that, we went out and measured the growth rate for those plants from tree cores and got the growth rate by mass scaling and compared it with our flow by mass scaling. And so here's the growth by mass scaling exponent. And you can see it's very similar to what we measured for flow by mass. This validates the original West Brown input proposition that you can predict growth scaling from flow scaling. So this validates the vascular covariance with productivity. And also the model, you can see, does a reasonably good job in both species. So this has been very exciting because it has validated a very important starting point, an assumption of this metabolic scaling theory, that growth is proportional to the transport rate. So we think that part we're quite excited about. Interestingly, the intraspecific scaling that we do predict does not get us to 3/4 power. Okay? So there's still this disjunct now between intraspecific scaling appears to be differing from interspecific scaling, which gives you 3/4. Okay? We cannot get 3/4 out of the model. So to the extent that Kleiber's rule really does, this 3/4 power scaling really does characterize interspecific scaling plants, it remains unexplained. So we're continuing this work because the model has given us so much encouragement. I didn't think it was going to be that accurate, but it's so accurate that we're continuing to refine it. We're particularly interested in the nexus between hydraulic architecture and productivity, so we're continuing to explore that. So that little bit of research is focused on how the flow resistance relationship of productivity scales with ontogeny and size. Now I'd like to switch to an evolutionary scale and talk about some work we've done on how flow resistance has evolved. You might hypothesize that as you go from very early vascular plants to more derived vascular plants, all those changes in the vascular system should be resulting in lower and lower flow resistances so that you can get more and more efficient plants. And there are extant plants around that reflect, serve as proxies for the different stages in that vascular evolution. And so we've gone about doing some comparative physiology to test this basic hypothesis that the evolution of the vascular system is related to reductions in flow resistance. And we know a lot about the evolution of the vascular system. The ancestral type of wick compartment here, xylem conduit, is called a tracheid, single elongated cell, a couple millimeters long, and the proper xylem is just a honeycomb of these tracheids. In the ancestral type of conduit, these compartments are called vessels, and they're not unicellular, they're multicellular, so they're a lot longer than tracheids. They tend to be lighter than tracheids, and they're interspersed in a more complicated wood anatomy. It's not all conduits, it's conduits, and then you have fibers and axial parenchyma. It turns out that vessels evolved from tracheids, and thanks to Bailey in a well-known and well-accepted manner. Here's the ancestral trachea here. In the evolution of vessels, the tracheids become short, and they lose those pitted end walls that are trapping the gas. So they just become little open, like toilet paper cylinders, called vessel members. And these vessel members are members of the vessel club because they stack on top of each other to make an entire multicellular vessel. So vessels are much longer, but they're not infinitely long. Where they end, they still have these pitted end walls to confine gas as is necessary. Now, in the evolution of fibers, they also evolved from tracheids by them becoming shorter and narrower. In more detail, you can see that the vessel evolution occurred in several stages. So this is the pitted end wall of an angiosperm tracheid, representing the ancestral state. And you can see these big sort of sheet-like structures. These are the pit membranes that are those microporous barriers between the tracheids that are trapping the meniscus. Okay? In the earliest transitional kind of vessel morphology, you lose the microporous barrier, and you're left with the supporting bars. And so you get these things called scleriform, because they look like a ladder, perforation plates. And the perforation plates are just the junctions between the vessel membranes. So that's kind of like the most transitional, primitive, if you could use that word, type of vessel element. And then progressively over more evolution, you lose these bars, and you get the most derived type of perforation plate. It's just an opening. And it's long been hypothesized that what's driving this is a reduction in flow resistance. But surprisingly, that really has never been measured in much detail. So we have done some comparative measurements on a lot of plants. This is to sort of test that hypothesis. This is kind of our sampling scheme. We have sort of an outgroup of which are the tracheids. Still like that. And I won't talk much about them today because I think Carmel's going to be here. And she loves to talk about conifers, so I'm going to let her tell you about those. But within angiosperms, we're looking at the evolution of vessels in angiosperms and their effect on flow resistance. And in the early diverging lineages, some of them still have the ancestral tracheid type. There's close relatives of those were selected that have that transitional vessel morphology. And in our third sample, we're late diverging angiosperms that have streamlined simple perforation plates. And so we just went around the world and we collaborated with Taylor Field on this, a great guy to collaborate with. To collect these early diverging angiosperms, they're not very common. They grow in very stereotypical kinds of habitats. And it was a wonderful sabbatic project. You got to travel to all these interesting places and sample these curious plants. And here are some of the results. Now, this is the conductivity of the xylem tubes per unit xylem tube area, so it's normalized, versus the diameter of the pipe. And the yellow here are the represent the ancestral state. These are tracheid-based angiosperms, or so-called vesselless angiosperms. And then on this graph, I've now plotted the other end of the evolutionary trend, these streamlined, highly derived vessels. And you can see, as the hypothesis would predict, there's a big jump even for the same width. The vessels are much more conductive because they're putting those pitted end walls further apart in space. And so that minimizes their contribution. So far so good. But of course, to get to this derived state, you have to proceed through these transitional stages, and those are preserved in the extant flora. And if you look at those, so early diverging angiosperms that have these scleriform type morphologies, you get a very different result. They're much less efficient than their eudicot counterparts with the simple perforation plates, and they're not really all that more effective than their trachea-based proxies for their ancestors. And if you normalize this by wood area instead of just conduit area, there's no difference at all between the ancestral proxy for the ancestral state, the transitional state in vessel evolution. It's only later in vessel evolution when you get rid of those scleriform perforation plates that you start to see a big advantage in terms of conductivity. So the implications of this survey were that the at least initial stages of vessel evolution really was not associated with a step change in hydraulic conductivity. Rather, it was essentially the same. These two wood types are about the same in terms of their conductive capability. So at least on a whole wood area basis, that's not a valid, this doesn't support the hypothesis that that was the reason that vessels were selected for. We think instead that the original sort of selected advantage of vessels was not to increase wood scale conductivity, but to keep it the same while making the wood much more flexible in terms of its other functions, leaving room for fibers to increase the mechanical versatility of the wood, and leaving space for parenchyma cells for greater storage capacity. And it was really the advantage of this more specialized heteroxylous xylem that was driving these initial stages, a much more subtle advantage. The tracheids have the efficiency of multitasking. This wood has the efficiency of specialization. So in this initial phase, our results suggest there was relatively little gain in flow resistance, and that the big gain came later in the later stages. Now, this implies that these scleriform perforation plates have a high resistance to flow. This bothered us because the literature on these plates, what little there was, suggests that they contribute maybe 5% to 10% at most of flow resistance. In other words, a fairly trivial effect. And these data came from computer models. So we thought, well, okay, we better check that out. And so instead of modeling it with a computer, we measured the flow resistance directly using the single vessel method that I think Brendan showed. You might check Sweineke were some of the first people to do this. Maggie Christman did this, where you draw out a capillary tube, stick it in a vessel, and then you can measure a single pipe, and make sure we have open pipes with no end walls here. So we just want to measure the flow resistance through the lumen. And Maggie did this on a whole set of simple plated vessels that derive type. And you can see, if you look at these vessels with simple perforation plates, for their flow, this is now resistivity, sorry to keep flip-flopping. And you can see as vessel diameters increase, the resistivity goes down, and it perfectly matches the predictions of the hydro-vascular equation. So the evolution of these simple perforation plates have essentially made the vessel lumen as good as it can possibly be as a conductor. Can't get better than the hydro-vascular equation, as far as I know anyway. So that's nice, but what about the scleriform perforation plated species? And Maggie did a survey of a whole bunch of those. And you can see, this is a log-log plot, so it doesn't look like a big difference, but actually it is a big difference. It averages about 92% or almost double the flow resistance. So these plates, far from just contributing maybe 5%, they actually double the flow resistance of the lumen part of the conduit. Way, way more than these modeling studies. We went back and looked at those modeling studies, and one of them modeled only one type of plate, and the other one actually had the morphology of the plates it's poorly represented in the model. We did a little bit of modeling ourselves, and our modeling actually comes much closer to this doubling effect. So these plates, removal of those plates was, according to these results, actually a pretty important event in terms of increasing the efficiency of the dusts. So you might be wondering, well, how come all these like obsolete xylem types with their low efficiency are still hanging around for us comparative physiologists to measure flow resistance? Minimizing flow resistance is so important. Why didn't they just get displaced by these streamlined vessels? And the answer is that not every habitat, flow resistance isn't equally important in every habitat. It's important in habitats where there's a lot of other resources, a lot of life, a lot of nutrients, where you can take advantage of conducting efficiency and outcompete neighbors. It's much less important in these kind of habitats where you find these plants, humid, nutrient-poor rainforests. They don't need high conducting efficiency. These kinds of xylems do the job just well. Now the conifers sort of solve the problem in another way. They actually are quite efficient, despite having tracheas, much more efficient than these guys. And that's partly because they have very sophisticated pits. They sort of evolve more efficient pitted involves. I'll leave that for the journal to talk about. All right. So that's sort of finishes what I can say about flow resistance and kind of the implications of that for plant performance. I'd like to turn now to considering the consequences of the strength of this hydraulic growth. And the reason that's an issue is because of the pressures that are generated by this wicking, this capillary suction inside the water columns. The pressures actually drop below the boiling point of water. So this is pressure below atmospheric log scale in megapascals. At sea level, water boils at about -0.1 megapascal. That's the boiling point. Measure xylem pressures, they're an order or two of magnitude below the boiling point. So the liquid doesn't vaporize, it stays, the molecules stay close together, and they're actually being put on under tension like they're a solid. And that's because of the hydrogen bond energies between water are so great that the kinetic energy of the molecules themselves is not able to break those bonds until you get way down to about -100 megapascals. At that point, the kinetic energy of these molecules is enough to break the weakening hydrogen bonds. That's the spontaneous failure of this metastable liquid phase. Now, of course, in real life, when we're boiling water, or pulling a vacuum on water, you do get vaporization typically near the boiling point. And that's because the hydrogen bonds are destabilized by nucleating agents, heterogeneous nucleating agents, of which bubbles are the most important, microbubbles. That's why when you're boiling water in a clean beaker, you throw in boiling chip, because you want to make sure that you've got nucleating sites so you don't superheat this water. The xylem just excludes boiling chips in their analogs, and thus allows this metastable liquid phase to persist. But, of course, the important question is, well, just how low can those pressures go? How stable is this metastable phase? How strong is this hydraulic growth? The water is I call it super sucked, because it's sucked below the boiling point, not heated past it. And that's an important question, and one that we've thrown a lot of methods at over the years. What would happen, okay, what happens is that at some point the pressure is going to get so low, you're going to get the nucleation of the vapor phase. This is called cavitation. And of course that's bad because the rope breaks and this vapor bubble will then expand until it's trapped by these capillary interfaces at the edges of the conductance. And the method that we use a lot of methods to measure cavitation in plants. The easiest to explain is one that Will Pockman put together when he was in my lab. You spin stems on a custom designed rotor, and the spinning generates a negative pressure in the central part of the stem. And if it's low enough, it will induce cavitation. And you get these emboli, and of course these emboli reduce the hydraulic conductance through the system. And so you can measure the hydraulic conductance as a function of the negative pressure, and as it starts to drop, you know that that's where the cavitation is occurring. And the result is what we call these vulnerability curves. In this one for sagebrush, the conductivity is plotted as a percent loss conductivity versus the xylem pressure. And the boiling point of water on this graph is about here. You can see that sagebrush is able to maintain a metastable liquid state down about -4 megapascals. And then you start to lose conductivity. There's some conduits that are more vulnerable than others, but eventually everything's gone in this species dynamic state. These curves raise a lot of questions, of course. Ecologically, the question is, well, how much variation do you get between species, and how does that relate to their adaptations to the environment? You get a lot of variation. You get really vulnerable species like cottonwood here, and really resistant ones. This is from work we did with Steve Davis. You know, this is from the local chaparral. Here's that sagebrush curve I showed you before. And as you probably guessed, or may already know, there is a pattern. These vulnerable species like the riparian cottonwood come from areas where the soil moisture is very predictable, and the only negative pressure is whatever is required to pull the water against friction and gravity, about a megapascal. Day after day, they just need a megapascal, and so they can afford, so to speak, to have a very vulnerable xylem, because they don't live in a place where they encounter more negative pressure. These guys living away from a water source, the soil will dry down, they got to pull harder to get water from a dry soil, they got to have a better xylem to do so. You can summarize this pattern on the next slide. We're going to plot the 100% cavitation point versus the minimum negative pressure observed in nature. And each point is a different species. This is the 100% cavitation point. This is the minimum pressure in nature, one-to-one line. Nobody's above it, of course. They wouldn't be able to do any gas exchange. But there's a strong correlation here. And this correlation explains a couple of things about the ecology of these plants. For one, plants like cottonwood cannot grow in these other kinds of habitats because they would experience pressures, as I just mentioned, that would push them over the line. So they're restricted by their vulnerability, or at least their vulnerability is consistent with them being restricted in that kind of habitat. What keeps these drought-resistant, cavitation-resistant species from going in and invading riparian zones? Certainly not cavitation. We suspect it's competitive exclusion, because the features required to develop these very high cavitation resistance are features which reduce the hydraulic conductivity of the zone and thus the productivity of the plant. So they would get out-competed in most habitats by vulnerable species. I won't talk too explicitly about those trade-offs, but basically there are trade-offs between resistance to cavitation and this flow resistance parameter, and also the density of the wood. The density of the wood is linked to it because of the need to support the conduits against implosion. By the fact, they're carrying water under suction pressure, so that unless they're reinforced, they'll collapse. Indeed, sometimes they do collapse. You might notice that we occasionally find some species on the line. In fact, the longer you sit around and observe the minimum pressure in nature, the tighter this correlation is going to get, because you're going to catch those 50-year, 100-year drought events, and everything's going to go this way in this graph. When you do get on the line, what happens? Well, of course, a lot of cavitation, and then also dieback and mortality. Not always the whole plant dies, but branches will die back, roots will die back, and sometimes the whole plant. I'm going to give you a couple examples of the nexus between cavitation and mortality. This one is a nice one. It's artificial, but it makes an important point, and it's an experiment done by Brandon Pratt in Albalac on cottonwood, which we saw was a very vulnerable species. And they raised this cottonwood in a humid greenhouse bag. The feedback loop between xylem pressure and stomatal conductance was never really entrained in this pampered plant, because it never encountered low humidities that required stomatal adjustments. So it was a lazy, stomatally lazy plant, and then they shoved it into a greenhouse bay that was very low humidity. Transpiration goes up through the stomata don't close because they haven't trained that feedback loop, and the pressures, of course, become more negative, and this happens, dieback by cavitation. This tells you one reason why plants might care about regulating their water status more than doing photosynthesis to avoid this kind of situation. That's especially the case in really vulnerable plants like cotton. Another example that's more natural is the pattern of dieback that we're observing in pinyon juniper forests in the western U.S. We've had a lot of chronic sort of failures of the summer monsoons, and that's been associated with dieback, selective dieback of pinyon, much more so than juniper, although junipers are also getting hit, but much lesser extent than the pinions. And underlying that, not surprisingly, are differences in their hydraulic equipment. Here, their vulnerability curves, pinions more vulnerable by quite a margin than juniper. Now, it turns out that pinion's not dying by cavitating to death, okay, like the cottonwood. Pinion is smarter than the cottonwood, and it closes its stomata at about -2, seemingly to avoid this upturn in cavitation. And it's very good at closing its stomata. So as a result, when the summer rains fail and the water potentials in the soil drop to -2, the plant has got its stomata completely shut. It is doing nothing. And for months, it will just sit there and do nothing. And it's obviously metabolically shut down. It becomes vulnerable to pathogens, which ultimately finish it off. So in this case, it's the avoidance of cavitation that is stressing the plant. Juniper, meanwhile, same habitat, much more resistant to cavitation. It will keep its stomata open and do gas exchange, albeit at low rates, but it stays metabolically active. It can do that because it is more resistant. It actually develops more cavitation than juniper, but it's tough. It can withstand it. Now, these two strategies, you might say, well, how come pinyon hasn't gone extinct? Well, pinyon is waiting for the summer rain. It's protecting its conducting system so that when the rains do come, you can suck that water up fast and efficiently, whereas the juniper is not only more resistant to cavitation and inherently less conductivity, but it's also may have experienced sustainable cavitation. So sort of a long-term, tough it out strategy, and kind of wait for to make hay while the summer rains come. Of course, every strategy has its trade-offs. Now, I won't tell you about it much, but we have a model. I developed a model that links these vulnerability curves to soil properties, the root shoot architecture stuff, and it can help predict these kind of stomatal response and dieback events. And we're working with Will Hocken and Nate McDowell in applying this model to a better understanding of this wide-scale selective mortality in pinyon juniper forests. And I think that model in general, it could be very useful for predicting plant responses in general to climate change. Just know a few parameters about the root shoot architecture. You can predict what the plant's going to do under different scenarios. Okay, but back to the vulnerability curve. There's other questions you can ask about these curves. Here's 3 of them from maple. And namely, well, what is it in the plant that's actually changing? So what kind of structural changes have to be selected for in order to turn a silver maple, which is very vulnerable, into a canyon maple, which is very resistant and, of course, grows in drier habitat? What is it that presumably the genes are dealing with that's bringing about these changes? We know a lot about this. Major cause of cavitation is the leakiness of these porous partitions, okay, which are supposed to keep the air out, but supposed to isn't always they succeed in doing so. And so let's take a look at the structure of these in more detail. So here's what it looks like in a micrograph. Here's one vessel, here's another vessel, here's this porous partition. Here this is here, we blow it up with SEM. It's holes in the wall, okay, but not complete holes because there's a pit membrane that spans those channels. It's not a cell membrane, okay, these are dead cells, this is just the primary cell wall. Okay, so it's a little porous partition that's there to let the water through, but of course it's there to trap the air-water interface and the inevitable event that you have gas in the system. And you have gas in the system even because of development. So the protoxylem vessels stretch, they rupture, they become gas. So there's no way the plant can prevent this from happening. Of course, they kind of leave breaks. Insect bites, you have air in the system. So the wicking forces establish this valve, but you can imagine as the pressures drop over here during a drought system, you're going to reach a point where that capillary force fails and the air propagates from one conduit to the next. So we can answer this question. Canyon maple has more airtight end walls than the acer saccharide and silver maple. But, of course, well, what is it exactly about these end walls? And an obvious hypothesis is, well, the sugar maple, this vulnerable species, ought to have pit membranes that are more porous. The bigger the pores, the weaker the capillary force. And that seems to be the case. So here we have a transmission micrograph across a pit membrane done by Frederick Lenz, who we collaborated with on this. The acer-saccharidum, the vulnerable species, has a very thin pit membrane, and you can see the pores in it. If you look at the canyon maple down here, a very thick pit membrane, and you can't even see any pores. And intermediate species are kind of intermediate. So adjustments in pit quality are part of the explanation. But there's more to it than that. So we've been doing a study where we are actually measuring the air seeding pressure. It's called air seeding, by the way, that leakage, of pitted end walls using, again, the single vessel method. And doing this on an oak. Excuse me. And here's what the data look like. So Maggie is spearing all of these big vessels. All of these are about the same size. And she's determining the air seeding pressure of all of them. And this, look at the distribution you get out of it. So this is the air seeding pressure. This is the percent. There is everything from incredibly leaky end walls down here that are leaking at less than a couple of bars pressure to all the way up to about 50 atmospheres of pressure and above. I mean, the capillary tube blows up and you can't measure it. And yet, they're the same size vessel. Okay, there's nothing different in the diameter. So to me, this reeks of probability. Okay, so there's a huge signal that there is a lot of probability going on in here. You can have some vessels that are incredibly resistant, and some vessels, a lot of them actually, that are quite leaky. You can see this in a more natural experiment. If you go out and measure the hydraulic conductivity for stem area as a function of natural xylem pressures in nature. You go out early in the morning, conductivities are high. You go out in the middle of the day, they're lower. You cut branches and dry them out a little bit more, you get this long tail out there. Again, you've got a population of very vulnerable xylem. You map that onto the predicted vulnerability curve from the air ejection data, and it maps on perfectly. So this is telling us that there are indeed a lot of really leaky vessels in there, and then there's some that are incredibly strong. And we think the reason for that, we think the role of probability is that in a big vessel, you have literally tens of thousands, if not hundreds of thousands of these pits that are being manufactured. And not all of these pits are going to be the same. Accidents are going to happen. You can see them if you look for them, where some of those pits are going to have bigger pores than others. And so simply because of this probability, and it only takes one hole to destroy the 100,000 pits. It's called the rare pit hypothesis, because the whole thing is driven by rare distributions, and that's what we think. There's our hypothesis for what's explaining this kind of crazy variation. Some of these guys avoid those mistakes, but some of them are inevitably prone to them. So there's two factors involved, to sum up, in determining vulnerability. One is the number of these pits through probability, and that's where the link between conduit size and vulnerability comes into play. Bigger conduits have more pits, thus more likely to have a weak bad pit in there, and so on. And then that's superimposed on adjustments in the individual quality of the pit membrane. So it's a complicated interplay between these two factors. Now, it's time for me to wind down here. I was going to tell you about our work on freezing stress, but I think I'll skip that. There are some also very nice trade-offs between conductive efficiency and vulnerability to freezing. Basically, I'll just give a take-home message. This is work that Steve Davis and I collaborated on. The loss of hydraulic conductance due to a freeze-thaw event increases dramatically in a step function with the mean conduit width, whether you're a vessel or a vessel angiosperm or a conifer or a tracheus. About 30 microns below that, you're okay. Above 40, you're completely cavitated. And it's interesting, if you look across the landscape, I'll just give you the latitudinal variation. You see that temperate trees tend to have narrower conduits, temperate diffuse forest trees, which keep their vessels going multiple seasons and thus have to go through the winter, have narrower conduits than tropical diffuse forest trees. And the y-axis here doesn't matter, just look at the x-axis. And it looks like the threshold, well, it's 30 microns. So again, you can explain this trend by selective pressures for the avoidance of minimization, freezing-induced cavitation in temperate trees. So we'll conclude at this point. Talk about how flow resistance through these pipes influences the productivity of plants, how their strength can influence survival, and a little bit of how trade-offs between these two are underlying the diversity and evolution of vascular structure. So I've got a few minutes anyway for any questions you might have. Thanks for listening. In the xylem vulnerability curve, what is the advantage of?
Yeah, what is the benefit of you curve it in a negative pressure versus a positive pressure? Because I copied out you have two curves that show the negative, and one curve is showing the positive.
Maybe it's because I'm plotting the conductivity as absolute conductivity versus percent loss. Is that what you? I guess I don't quite. So you can do a vulnerability curve. I hate these. I got it in my lap. Anyway, you can if you plot a vulnerability curve with conductivity, instead of percent loss of conductivity, it goes from high to low. When you plot it as percent loss, it goes from low to high, but it's the same curve. Yeah, it's just plotting it. Sorry. I actually like to plot them in absolute conductivities because I think there's more information there, and I also think it's more accurate because the percent loss, you're taking every conductivity value and normalizing it by one value. And if that one value is changing or drifting, you're screwing up all your data points. Whereas if you just plot it as absolute conductivity, maybe scale for stem area, I think it's more informative. I think that's what you're asking.
So about your allometric scaling model, so because C4 plants are much more water use efficient, do they scale differently then?
I guess you would predict in the model. See, the model doesn't predict absolute growth rates. The numerical model does seem to predict absolute water use rates as well as scale. But it only, at this point, only predicts growth rates scaling. So the simplest hypothesis would be the C3 and C4 would have similar scaling, but the growth. But if you could predict, and I hope to see the model incorporate carbon allocation so we can get water use efficiency out of the model, you have to take the flux, water flux that's produced by the model, couple that to the assimilation, and then partition it out into respiration, blah, blah, blah. And then you could predict the absolute growth rates as well as how they scale. So I guess we predict they scale the same, but the intercepts would be different. Yeah.
If these things were all at such strong negative pressures and they might cavitate more or less spontaneously if there was something else aside from a condensation nucleus that was present, if you walked around with an ultrasound generator, could you kill a bunch of trees? Yeah, well, the guy who first observed cavitation in plants that I know of, maybe somebody before, but nothing's new under the sun or whatever, but John Milburn, and he developed back in the 60s, early 60s, and he developed an acoustic device to detect cavitation by the energy put on. And he did a bunch of experiments bombarding the xylem with ultrasonic waves, to see if that would stimulate cavitation. And essentially everything that he did like that had no effect. There was no effect.
So it doesn't look like you could go and shoot gamma rays at the tree and kill your neighbors.
Pete Scholander.
Yeah, well, Scholander he did not, to my knowledge, study cavitation or postulate it. But he was the guy that, along with Hamel, developed the pressure chamber for actually measuring these negative pressures.
So are there examples of developmental changes from seedlings to mature plants in these xylem traits?
Yeah.
Seedlings often being very much more stressed.
Well, I don't know about related to that, but certainly Brendan Choate has shown that primary xylem tends to be more vulnerable than secondary xylem, like within a single species, and that, he's demonstrated that directly. And the explanation for that is the primary xylem has a lot more intervessel pitting than the secondary xylem, and thus by probability it's going to be more susceptible. So there may be some kind of a developmental constraint, maybe, if you want to use
Thinking
that.
about shallow-rooted seedlings that might
Yeah.
not need
Now
to
that-
the water supply yet.
Yeah, we've done that, like with juniper, we compared juveniles versus adults. We thought, well, maybe the juveniles would be more resistant. And not a whole lot of a difference there. They're a little bit different, but not stunningly so. Yeah, so nothing general that I can say.
We found the same with Randacy.
The same, not much of a difference.
So your upper limit for tracheids and vessel size in response to freezing is the same, but your xylem density is lower in your plants with vessels. Like, so you'd expect to see like less flow for angiosperm. Okay.
Yeah, well, actually, that's one of the equalizers between temperate conifers and temperate angiosperms in terms of their flow conducting. They actually, conifers do, whether you measure them or predict them with that model, they're side by side in terms of their conducting efficiency. And that's for two reasons. They have, like you said, they have more tracheids per unit area by a long shot. And their intervessel pitting is 60 times more efficient than the angiosperm intervessel pitting. So they've found ways to make a trachea-based system competitive, at least in a temperate, or probably the same would go on a nutrient-poor tropical kind of a habitat. And again, there are selective pressures for, sports car xylem is not there. Okay, well, thanks for listening to xylem for 15 minutes.